Let us study a new chapter, Heredity and Evolution. What is meant by heredity? Heredity is the transmission of biological characters from one generation to another. Example, characters from grandparents to the parents, again from parents to the young ones. So it's a transmission that is known as heredity. Now, what is meant by evolution? Evolution means change in the characteristics of a species over several generations and depends on the process of natural selection. Evolution is occurring only because of nature's selection. We can say that we human beings formed from monkeys to apes, again from apes to human beings. In human beings itself you can find that Neanderthal man, Cro-Magnon man, then at present modern man, that is Homo sapiens. So there all we can find that so much changes. These changes are known as evolution. Now, for occurring evolution, there takes lakhs of years. Once a change occurs, that change remains as permanent. Now we will study about different functions of DNA. First we will study what is DNA. Now, first I am showing you cell. Inside the cell, there is nucleus. Inside the nucleus, there will be chromosomes. These chromosomes are made with the help of DNA. Now, here inside the DNA, there will be thousands of genes. Now, genes are having a specific function. One gene has to do one function, that is for making either proteins or enzymes. At the same time, in case of DNA, there will be thousands of genes. Then each genes are having specific function. Now, here we are going to study about DNA as well as RNA. DNA helps in the formation of RNA. RNA itself is of many types. Out of them, we are studying only about three main types of RNA. They are mRNA, tRNA and rRNA. mRNA, that means Messenger Rna. tRNA, T, that is transfer RNA. Then rRNA, that is ribosomal RNA. Now, what is the full form of DNA as well as RNA? DNA, it's a full form deoxyribonucleic acid. Deoxyribonucleic acid. RNA, it's a full form ribonucleic acid. DNA, it's a full form deoxyribonucleic acid, and RNA, it's a full form ribonucleic acid. DNA helps in the formation of all types of RNA. Here we are studying about these three RNA, messenger RNA as well as tRNA, these two, messenger RNA as well as transfer RNA producing inside the nucleus, and ribosomal RNA is producing inside the nucleolus. Now we are studying about the production of messenger RNA by DNA. So at first DNA produces messenger RNA. This is happening inside the nucleus. This process we can say as transcription. Transcription. So conversion of or production of messenger RNA with the help of DNA, that process is known as transcription. Once messenger RNA will form, messenger RNA already formed now, this thing slowly enter into the nucleolus, into the cytoplasm. This is nucleus, this is cytoplasm. As and when messenger RNA produce, slowly it will enter into the cytoplasm. Now once it will form, it will enter into the cytoplasm, it will reaches into the ribosome. There the production of protein will take place. So we can say that messenger RNA helps in the production or in the synthesis of protein. So DNA helps in the formation of messenger RNA. As and when messenger RNA will form, that messenger RNA release out from the nucleus into the cytoplasm. There it will help in the production of protein. Now inside the cytoplasm there is ribosome. The main function of ribosome is protein synthesis. Now here messenger RNA is reaching, similarly transfer RNA as well as ribosomal RNA, all these RNA will be present in the ribosome. They do different functions. Now messenger RNA, we studied that it is helping for the production of protein. Transfer RNA is helping for the transfer of amino acid in the production of protein. Ribosomal RNA is helping in the formation of framework of ribosome. Now as and when messenger RNA's production happen, it enters into the cytoplasm. Now how the transcription process is taking place, that first we will study. After that messenger RNA, how it is entering into the cytoplasm, that also we will study in detail. Now which process I am showing you, that is happening inside the nucleus, that is transcription process, means formation of messenger RNA, that is mRNA, from DNA. Now DNA is having such a structure, we can say that it is having a double stranded structure, or we can say that it has a double helix structure. So this is the structure of DNA shown. We can say that it has double stranded or double helix structure. Now we can say that here two ribbon like structure it has. again inside we can say that like that way bonds will be there. Such a way bonds will be there. Hydrogen bonds we can say. Plus it is highly coiled structure. It has so much a coil is there. Very lengthy it is. Then we can say that it has such a bonds are there. Again we can say that here where bonds begin as well as ending, there nitrogenous base is there. Now there are 4 different types of nitrogenous bases there. That is known as adenine, guanine, cytosine, then thymine. So here we can say that adenine, then guanine, cytosine, then thymine. Like that way, here it's a short form A, G, C, then T. Now here one more specialty is there. Here A as well as T pairs, joins, as well as G as well as C pairs, joins. So how it will be? Here now suppose A I written T, G, C. Like that way number of, we can say large number of such a nitrogenous base will be there on both the strands. Here I written ATGC now. Then its opposite, here A is there, then here will be T. That is pairing work, AT. Then here T is there, then again A. Pairing. Now G is there, then C should be there. GC. Now C is there, then here G should be there. So such a way pairing work is happening inside the DNA. Now such a way means here all it will be there. It is such a highly twisted or coiled structure is there. We can say spring like structure it has. In that large number of such a nitrogenous base or we can say nucleotides will be there. Now this both of them join with the help of a bond. This bond is known as hydrogen bond. Now where formation of messenger RNA is taking place, or we can say DNA transforms to messenger RNA. That process is known as transcription. So we can say that conversion of or formation, conversion not formation of DNA to messenger RNA. That process is known as transcription. At a time, everywhere transcription won't take place. Where transcription takes place, that particular place, we can say that DNA unwinds or untwisted condition will be there. Remaining place, same way formation will be there. So where messenger RNA forms, that particular place, we can see that this strand will be lying in the parallel condition. Other place is same to same condition you can see. Now that figure I am showing you. Here now where formation of messenger RNA, that place I opened and kept. Remaining place you can see that like that way. Twisted condition. Similarly here. Now here only the process of transcription is happening. Transcription means formation of mRNA from DNA. mRNA is a full form messenger RNA. This process is taking place inside the nucleus. Here now again I am making the nucleotides or nitrogen base. Like that way, there will be large number of nitrogen base will be there in one strand or one space, we can say one coil, one spring like a part. Many coils are there. In one coil itself there will be large number of nitrogenous base. So I am writing again nitrogenous base only 4 types, A, T, G, C. A means adenine, thymine, guanine, then cytosine. Again now cytosine, guanine, thymine, adenine. Like anyhow we can write. Now any order, any sequence we can write. Now I am writing A, then T, any. Now but take care that here now A is there, its opposite side there will be T. Here T is there, then A, G is there, then C, C is there, then G, C is there, then G, G is there, then C, T is there, then A is there, then T, A is there again T, T is there, then A. So that means a matching pair. That means a pair A can form pair with the T only. C can form pair with the only G. Now these nucleotides or nitrogenous base join with the help of a bond. This bond we can say as hydrogen bond. They join with the help of a bond. That bond is known as, it should be in straight line. This bond we can say as hydrogen bond. So with the help of this hydrogen bond, these two nucleotides or we can say nitrogenous base joined. Now when messenger RNA will form, that particular time this bond will break. This bond will break. Then here we can find that other nitrogenous base will come and join, which is already there in the nucleus. Inside the nucleus large number of nucleotides will be there or nitrogenous base will be there. So here already bond broken now. So there you can find that new, here now A is there, then T is there, A. These all nucleotides will come and join from the nucleus. Here G, here C again G, G then C, then T, that is A, then A, that there T, then A, T, T, here A. Now we can say that here in between them, so such a way new nucleotides came and joined here. So before that what happened? This hydrogen bond already broken. There other nucleotide came and joined. Now these nucleotides will join together in the form of a chain. Now these nucleotides came and joined here with the different nucleotides. After that these nucleotides joined in the form of a chain. Then we can say that this is known as a new messenger RNA. So here new formation of the RNA took place. Then these all hydrogen bond will disappear. You can find only messenger RNA here. This is known as now messenger RNA. One strand, new strand formed. the newly formed strand we can say as messenger RNA. Then this messenger RNA slowly will come out from this strand or this coil and it will enter into the cytoplasm. So here now we can say that this is known as messenger RNA. As and when the formation of messenger RNA will take place, it will come out from this DNA and from nucleus and it will enter into the cytoplasm. Now here this part is coming out that I am showing you here now, that is T, A, C, G, again G, C, A, T, T, A. Now here one correction is there. When messenger RNA will form or RNA will form, in RNA there is no T, no thymine. T means thymine. T won't be there. Instead of T, there will be the formation of U. U means uracil. Uracil. There will be the formation of uracil. T won't be there. Instead of that, there will be other nucleotide. That nucleotide is known as uracil. So here T I am removing. I am writing here U. Here again I am writing U. Here again I am writing U. Remaining nucleotides will be same. Now we will study further what is happening. As we have studied, messenger RNA is helping in the production of or synthesis of protein. Now how the protein synthesis is taking place in the messenger RNA, that we will study. Now here I am drawing messenger RNA. This messenger RNA will have many joins. Pieces joined like it will be there. Then messenger RNA brings messages from nucleus into the cytoplasm. So messenger RNA carries messages from nucleus into the cytoplasm. Now here this messenger RNA will have such a sequence will be there, such a structure will be there, such a structure will be there. We can say as codon, like that way structure will be there. Each such a sequence carries one nucleotide with it. So here now, in case I am writing A, T, G, C. Now here again I made the mistake, that is, this is the mRNA. mRNA. In case of RNA, there should not have T. Instead of T, there should be U, then C, then G, U, A, G, C, U, U. Anyhow, anyway I am writing now U, G, C, C, U, G, U, A, U, G, U, A, then U, G, C, C, A, U. Like that way any letter I written. Now here we can say that this particular place now one amino acid. That means here this messenger RNA has codons are there. We can say that 3 nucleotides together, one codon is becoming. That means one message will be there. This message for amino acid, which amino acid want to join with it, for that. So here we can say that 3 nucleotides join together, one codon is becoming. So this is known as triplet codon. Triplet codon. So here we can say that this messenger RNA came from the nucleus. Now it is lying inside the cytoplasm. Now here this messenger RNA carries large number of nucleotides with it. We can say that nitrogenous base with it. Now each one 3 nucleotides together, one codon is becoming, CODON. 3 nucleotides together codon is becoming. So it is known as a triplet codon. Now each one we can say as each letter. That means each nitrogenous base we can say as codon. This is known as a triplet codon. Why? 3 nucleotides together, one amino acid can be joined. So here now total there are 20 amino acids. In the nature or in the living things, altogether 20 amino acids are present. In English language, total 26 alphabets are there. With that, large number of words we are making, innumerable number of words. Similarly, with the help of these 20 amino acids, total 20 amino acids only present, not more than that. So in the nature, total 20 amino acids are there. By joining these amino acids, innumerable number of proteins can be made. So here, which, like that way, triplet codons we are making now. Like that way, triplet codons are making. Now, last I am making U, G, this I am erasing, A. So here again I am making the sequence codon, we can say. Now here the last. So now such a way 3 triplet codon I made here, any sequence, but one thing we have to take care that start codon should be AUG. Then stop codon can be UGA or UAG or UAA. Out of this, any. So ending place, once the process will end, one protein's production will over. So end codon or stop codon will be there. Beginning place start codon will be there. Now start codon should be AUG, end codon should be either UGA, UAG or UAA. Now here in this particular place, now in this place AUG I written, sometimes it can be other also. Any other I am writing? CAG. C means cytosine, adenine and guanine. This is other one. So here now I am writing some alphabets here, the word form. Like that way one word I written. So here now you can see all alphabets I joined and made one word. Now when we read that, here any meaningful word is there or not. So like that way when we read meaningful word we have to make from this number of letters which is written here. So when we see here like that way we read and go, we will find here, we will find SOME, one meaningful word here. So we started to read from here and reached here we got one meaningful word. Similarly, here when the amino acid is coming and amino acid is transporting with the help of other RNA, that RNA is known as transfer RNA. So here now already this messenger RNA is lying in the cytoplasm. Now already it is reached in the cytoplasm. Now here one transfer RNA will come. Transfer RNA that is having almost we can say that such a structure will be there. A special structure will be there. I am making now. Such a structure, approximately such a structure will be there. There it will be having 3 anticodons. Here now codon is there, codon, triplet codon, 3 letters are there. Now here 3 letters will be there, anticodon. So here now in this particular place, like that way keep on reading and going, this is a transfer RNA. So when it is going, now suppose here AUG is there. AUG means here A for U will be there, U for A will be there, G for C will be there. AT. As we have studied here, A pairs with the T, G pairs with the C, C pairs with the G, then T pairs with the A. Here now, in this place, this is the RNA, so there T won't be there, there will be U. Here T won't be there, here U will be there. So here now, we have to see that from here reading begins. With the help of, here ribosome will be there, transfer RNA will be there. So inside the ribosome there will be RNA. So we can say that here this transfer RNA will be along with the ribosome. It will come towards here. Afterwards it start to read where suitable codon is there. So here now this is known as the transfer RNA carries codon. This codon is known as anticodon. Again, this end region will have amino acid. this is known as a tRNA. tRNA will have anticodon as well as it carries amino acid with it. Then here along with it you can find that a ribosome. Why? Because RNA, which is helping for protein synthesis, that is messenger RNA. Again we can say that this tRNA is having the duty to move the amino acid with it. Now again we can say that first it goes to this transfer RNA transfers amino acid, but it will be along with the ribosome, and this transfer RNA helps in the protein synthesis. So messenger RNA doing protein synthesis. Now to the messenger RNA, which all help required for the production of a protein, that is doing by transfer RNA. Now here we can say that the protein synthesis begins with the start codon, that is known as AUG. So from here, now suppose this transfer RNA came here. So this transfer RNA along with the ribosome start to move through the messenger RNA and start reading this codon. Now here CAG became, so it found that it is unwanted. Why? Because starting codon should be AUG. This is not the starting or start codon. Start codon always should be AUG. So this transfer RNA along with the ribosome start to read the codon as and when it reaches AUG. So we can say that this is the start codon. Start codon. Now this start codon, as and when it reaches, immediately it comes towards this. It comes towards here. That means AUG is the start codon. Now these three anticodons. So A is there, we can say U is there, then A, G is there, then C. It will come and join here. Now, as along with that, it brings one amino acid. So AUG is there. For this anticodon, UAC is there. Again, it brings this UAC and transfer RNA brings the amino acid. So for this start codon, one amino acid is there, that is known as methionine. Methionine. This is the starting stage which amino acid want to attach. That amino acid is known as methionine. So here now, after this amino acid is coming close to this start codon, this transfer RNA will disappear. It will disappear. There will be the formation of amino acid. Now this amino acid I am showing you here. It will come close to it. Again other transfer RNA will come. This will disappear. First one already came and kept one amino acid here. Now again keep on reading by the ribosome. At the same time again there will appear one transfer RNA. Now there will be again anticodon. Now which will be here, now as and when we read, we found here a meaningful word. Same way here, this ribosome keep on reading, it will move. where it is finding a meaningful codon, there it's a matching anti-codon will come and join. Suppose here as one AUG over, now here CGU is there. Its matching anti-codon will come and join here. Along with that, there will be the formation of one amino acid. Formation not, there will be this transfer RNA will carry a suitable amino acid along with it. Then here also anticodon will be there, any letter may be there. I am writing here now C, G, A. One anticodon I written here. Now matching to this, where it is there, it will search, it will read. Where C is there, so that means that G should be there starting. G here it is there. At the same time G, for that C is there. Then here but C again is there, but here what should be? A is there, that means U should be there. So for this suitable codon, this is the anticodon. Transfer RNA brings anticodon. Transfer RNA brings anticodon. So now here this is the amino acid, methionine, that is the amino acid. Like that way total 20 amino acids are there. All names by hearting is very difficult is there. Now even lysine, one amino acid is there. Then we can say glutamine, valine, serine, proline, then tryptophan. Like that way different types of tryptophan. Tryptophan, not tryptophan, tryptophan. Tryptophan. Like that way different types of amino acids are there. Total 20 amino acids are present. Now here CGA, anticodon, is moving now for searching or reading where suitable triplet codon is there. For seeing that it is searching. So now C is there, C will be there, G should be the starting letter. Where G is there, here G is there, then G is there, then C should be there, that is also there. Then A is there, but here C is there, so no, cannot. Now like that way here C is there, G is there, afterwards G is there, C is there, but A, then it should be U, not there. So somewhere will be its matching codon here. So there it will go and it will attach with the codon. Now just I am changing it, but the change we cannot make it. It is already forming from the DNA. So where it is there, there this thing will go and it will attach. So here now CGA is there. So here nowhere it can. So again no way is there. So what will be, where it is there till the place it will join. So here again I am making different one. G, then here C, afterwards A, now I made here now it's a matching one, but there will be in the messenger RNA somewhere it's a matching will be there, matching codon will be there, there it will come and attach. So here now CGA is there, so for CG its a matching should be G, C means G, then G means C should be there, A means U should be there. So GCU where? It will keep on searching like that, then finally it will find here. In this place also we can find GCU, but the problem here 3, again 3, again 3, like that with 3 triplet codon will be there. It's a matching GCU. We can find now only GC here. U is there, that means A is there here, then there should be U. Like that. So here now C for that G, G for that C, A for that U. So here G, C, U, here it is finding. Then what will do? This will keep on moving, reading, where suitable one is there, here it is finding. So again it will form its anti-codon here. Then it will bring and keep its amino acid here. Then once here now I am removing. Once here one amino acid kept. Now this place one amino acid kept. Here we can say GCU. Then amino acid keeping work over, it will vanish, it will disappear. Now again other transfer RNA will come, that is tRNA again will come again to it, which is the suitable codon. It will search, it will come with the amino acid. Now suppose here now in this particular place AC, here it's straight, it will keep on reading, it will come and stand here where it's suitable anticodon is there. So it is here now A is there. A means there should be U, then G, because this is a RNA, so instead of T there should be U, G is there, C is there, then G. So I am making here now like that way, U, C, G, and here one suitable amino acid will be there. So once it is coming and after reading it is finding that suitable codon is there, then this anticodon will fix here, anticodon will fix here, then it will leave the amino acid here and the transfer RNA will disappear. So same process continue till when? Till it finds, this is known as stop codon. Stop codon. Stop codon, that is either UGA or either UGA, or we can say UAG, or we can say UAA. So like that way UA here. Now you see here also it is there UAA. But problem what is there? Here UGU one triplet codon, AAG one triplet codon. So there means nothing is possible. Alone in one triplet codon, here UGA. This is the triplet codon. Here UGA is there. As and when it come here, that means the transfer RNA here it reaches with a suitable anticodon. That is a UGA. It's a suitable anticodon will be there. That is A, C, then U. Then work is completing. So till it is reaching, as and when it reads, different tRNA will come, or many tRNAs will come to search the matching codons. As and when it finds the matching codon, it will come and fix the amino acid there, amino acid in that particular place. So like that way suitable place amino acids will bring and keep by the transfer RNA with the help of ribosome. Now such a way reading and coming and fixing the suitable RNA as well as anticodon by the transfer RNA, that is known as translation. So before we studied about transcription, transcription is the formation of messenger RNA by DNA. As and when messenger RNA form, it carries coded message from the nucleus to the cytoplasm. As as and when it reaches in the cytoplasm, it will have many coded information with it. The coded information with it will be in the form of triplet codon. Now, once it will come into the cytoplasm, for each triplet codon, matching anticodon will bring by transfer RNA. And this transfer RNA read all the coded messages with the help of ribosome, and where suitable place is there, that suitable place, this transfer RNA bring and fix 1,1 amino acid. Till this process will be continued till it finds one stop codon. Now, this amino acid fixing process will start from the start codon. Start codon is AUG. Stop codon will be either UGA, or we can say UAG or UAA. There, once it reaches to the stop codon, that is UGA, UAA, or we can say UAG, there the bringing and keeping or fixing amino acid function will stop. Now, the brought amino acids will join together and will form. That means amino acids will join together, a protein will produce. So here we can say that this messenger RNA is helping for the production of or formation of a protein. So here now we can say that amino acids join together. Amino acids join together forms proteins. Amino acids, like that way now all these amino acids join together, one protein is formed. Similarly, this process, once the protein formation will take place, then this messenger RNA will disappear. tRNA, whenever anticodon's necessity is there, then amino acids fixing work is there, that particular time new transfer RNA will come along with the anticodon, and suitable place, after reading where its suitable place is there, it will bring and keep anticodon as well as amino acid. As and when stop codon reach, the amino acid fixing work will stop. Even anticodon's fixing work will stop. Then how much amino acids formed? Its a chain will form. Then as a result of it, there will be the formation of one protein. Now, as and when protein form, this messenger RNA will disappear. Again, new messenger RNA can be produced with the help of DNA as and when there will be the requirement of formation of new protein. So, DNA is having the capacity to produce messenger RNA as and when required. Once the function of messenger RNA completes, that messenger RNA will disappear. Then as and when again protein's necessity is there, protein synthesis is required, again new messenger RNA will be produced by the DNA. So the production of messenger RNA by DNA, that is known as transcription. Now afterwards, fixing of anticodon as well as amino acid wherever necessary, that is happening with the help of transfer RNA as well as ribosome after reading it. That process is known as translation. As and when protein synthesis is completes or protein synthesis happens, this messenger RNA disappear. As and when new proteins requirement is there, new protein synthesis is required, that particular time again DNA will produce a new protein, new RNA, that is new messenger RNA. Then as and when this new messenger RNA completes protein production, a new protein production, again new messenger RNA disappear. Again after some time protein synthesis is required, then DNA produces new messenger RNA. Then with the help of that messenger RNA, new protein will produce by taking the help of tRNA as well as ribosome. So this process continued in the living things inside the nucleus as well as cytoplasm. Messenger RNA produces inside the nucleus. At the same time, the formation of protein is taking place inside the cytoplasm in the ribosome. Each messenger RNA has thousands of triplet codons. As per the message on the messenger RNA, transfer RNA, that is tRNA, supplies amino acids to the messenger RNA. This is known as translation. Translation. What is meant by translocation? During the process of protein synthesis, ribosome moves from one end of mRNA to the other end of mRNA by the distance of one triplet codon. It's the meaning. Here, this, just imagine this is the ribosome. Now, this ribosome, this is the messenger RNA. This ribosome moves from this, through this messenger RNA from one end to the other end. Now, at the distance of one triplet codon, here now one triplet codon, again it will move and it will reach up to here. Again it will move, again it will reach up to here. Like that. So we can say that during protein synthesis, that means a messenger RNA, its function is protein synthesis. During protein synthesis, this ribosome moves from one end to the other end of messenger RNA or mRNA. Now at the distance of one triplet codon, now this is one triplet codon, again it is reaching here, other triplet codon, again it is reaching here, other triplet codon, again it will reach, it will move like that, it will cover one triplet codon, again it will move, again it will cover one triplet codon, such a way. So we can say that it will move from, that means during protein synthesis, ribosome move from one end to the other end of mRNA at the distance of one triplet codon. This is known as translocation. So we have studied here now transcription, translation and translocation. What is meant by transcription? Transcription means formation of mRNA from DNA. What is translation? Each mRNA has thousands of triplet codons. As per the message on messenger RNA or mRNA, tRNA supplies amino acid. This is known as translation. What is meant by translocation? During protein synthesis, ribosome moves from one end of mRNA to the other end of mRNA at the distance of one triplet codon. This is known as translocation. What is meant by central dogma? Information about protein synthesis is stored in DNA, and synthesis of proteins is very necessary for the body. These proteins are produced by DNA through RNA. This is known as central dogma.